Hello students, in today's class, we are going to learn about genetic code and its properties. So basically, this slide represents the basic structure of amino acids, and each amino acid basically consists of a central carbon to which 4 different groups are attached. One of it is always hydrogen, another one is an amino group, a carboxylic group, and an R group. So depending on the type of R group, amino acids can be categorized or classified into 5 different categories. So these are examples of some of the amino acid structures. So during protein synthesis, we basically have peptide bond formation between carboxylic group of one amino acid and amino group of another amino acid, forming a CONH bond, which is known as a peptide bond. Okay, so protein synthesis is brought about by ribosomes, which is the protein synthesizing machinery. During translation or protein synthesis, messenger RNA is translated. It is basically read in 5' to 3' direction, producing a corresponding N-terminal to C-terminal polypeptide chain. During this process, amino acids are bound that are bound to the transfer RNAs are inserted in the proper sequence because of specific binding of each amino acid to its transfer RNA and specific base pairing between the messenger RNA codon and transfer RNA anticodon. So next, we are going to discuss about properties of genetic code. So genetic code is basically made up of 3 bases that form a single codon, also known as a genetic code. So since there are 4 bases, that is C, A, T, and G, so at a particular location or a position, we can have 4 possibilities, that is any one of the 4 bases can be present. Similarly, at the second position of the codon also, we have 4 different possibilities, that is either an A, G, C, or T is present. Similarly, at the third position. So because of this, the product of 4 times 4 times 4 is 64, and hence we have 64 different codons because of different combinations of the 4 bases. Out of these 64 codons, 61 codons are sense codons, and 3 codons are known as termination codons. Next property of genetic code is that it is a triplet, that is, it is made up of 3 bases. Next, it is degenerate in nature. This means that a particular amino acid can be coded or encoded by more than one triplet codon. For example, in case of lysine, we have 2-fold degeneracy as it is encoded by 2 codons AAA and AAG. Next property suggests that genetic code is non-overlapping. So here is an example of a partially overlapping codon wherein CAT is forming the first codon and TGA is forming the second codon. So here the thymine base is shared between the first and the second codon. So this is an example of an overlapping codon wherein one base is being shared by 2 codons. And in the second case over here, we can see that CAT is the first codon and ATG is the second codon. So here is an example where in 2 bases, that is A and T, are shared between 2 codons, CAT and ATG. Okay, so this is an example of overlapping codon. However, this is never the case. This is what is explained in this property, that genetic code is non-overlapping, means that one particular or 2 bases cannot be shared between 2 codons. Next property is genetic code is commaless. So this basically means that if suppose CAT is forming one codon, then there is no allotment of any base that will indicate the end of the first codon and the beginning of the second codon. So in the sequence, if CAT is the first codon, automatically the next three bases will form the second codon. So here is an example where they are showing a random base X is being used as a punctuation or as a indicator of end of the first codon and the beginning of the next codon. But this is never the case. Always, we don't have any base that is used as a punctuation or as a comma to indicate the end of the first codon and the beginning of the next codon. So this basically means genetic code is commaless. Next property is genetic code is non-ambiguous. This means that the same codon does not codes for more than one amino acid. For example, if AUG is encoding for methionine, it cannot encode for another amino acid such as lysine as well. It will only encode for one amino acid, that is methionine. Next property is genetic code is universal. This means, with the help of an example, if AUG is encoding for methionine in bacteria, then it will encode for methionine in eukaryotes, in viruses, in all the living systems. Okay, next property is its polarity. Genetic code is always read in 5' to 3' direction, as is your messenger RNA. Next is the chain initiation codons. So there is one initiation codon read in 5' to 3' direction as AUG codon, which encodes for methionine amino acid. In case of prokaryotes, it is a formaldehyde methionine that is encoded by the initiating AUG, while in case of eukaryotes, it is an unformaldehyde methionine. Last property are the termination codons. There are three termination codons, UAA, UAG, UGA, which will signal for end of the translation process whenever they are encountered by the ribosome. So that's it about properties of genetic code. Thank you.